Hello, everyone, and welcome to today's webinar, Revolutionizing Pharma, How Intelligent Automation Accelerates R&D and QC Inspection Processes. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Galatech. To learn more, visit galatech.ai. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd like to now welcome our speakers for today's webinar. Dr. Yicheng, Senior BD Director at Galatech, and Dr. Yuyan, Application Scientist at Galatech. Dr. Yu, you may now begin your portion of the presentation.
Hello everyone, welcome to this webinar, and my name is Yicheng Liu. I'm the General Manager of Galatech European Division. And within today's webinar, we will specifically introduce the solutions that we designed for the pharmaceutical industry. For example, the compound synthesis platform, the small molecular drug high throughput platform, and also the QC inspection mobile phase preparation system. And however, before I dive into today's solutions and automations, I would like to give you a brief introduction about our company. So the headquarters of our company is in Singapore, and in the beginning of this year, we just entered into the market in Europe, and now we are establishing the demo center and also the R&D center in Munich. And globally, we also have presence in Japan and also in the US. And of course, the majority part of our company is in China. So within China, we have R&D delivery center factories in all the mega cities, and actually the China division's headquarters in Suzhou. And since the establishment of our company in the year of 2016, we have been providing advanced automations for life science, healthcare, and also drug discovery. And some quick facts about our company. So at this moment, we have employee more than 1,000, and around 60% of our employees have the R&D position, and also in the meantime, 60% of our employees have the master or above degree. And through this number, I just want to demonstrate that our company looks at the R&D development, the technology evolution very importantly. So we are determined to invest significantly in the R&D process. The core business of our company is life science solution. So at this moment, we are to build up integrated platforms for high throughput drug screening, omics sample pretreatment system, organoid culture system, synthetic biology platform, and platform for cell line development. And traditionally, we look at the drug industry very importantly. And in the next slides, I would like to explain our company's vision and also perspective for the drug discovery industry, and also our mission for the life science infrastructure revolution. So as we know, the development of new drugs divided into several different phases. For example, the early phase drug discovery, the preclinical and clinical research, and also the regulatory approval. And in average, the success rate for phase 1 drug to get the regulatory approval is only 7.9%. And with average time required of 10.5 years. And this is definitely not a sustainable way and efficient way for the drug discovery. To further improve the efficiency of the drug development, some long-standing models have to be disrupted, and the pharmaceutical companies need to find new ways to compete and win. So there are two primary strategic pathways for the future, and one is based on the automation and AI solution, and also the other strategies based on the quality and scalability. And at Galatech, we believe the AI and automation will revolutionize the drug discovery, and the combination of automation and AI will primarily accelerate innovation, enhance quality, and provide scalable solutions. And the solution that we want to provide to support the lab science infrastructure revolution and is wide and dry lab combined solution. So by using our solution, we can design the experiment, edit the workflow, and also generate, archiving, analyze, and manage the data. So for example, we can provide chemical feeding and chemical reaction solutions for the small molecular drug discovery, and we can also provide 3D disease model, culture and screening system. So this is specifically designed for high throughput drug screening. And for the QC inspection process, we can also provide mobile phase preparation system. And last but not least, we can also provide intelligent chromatography testing system so that we can connect different functional units or even different functional labs to realize the whole workflow automation. The first automation, the first solution that I want to dive into today is the high throughput chemical feeding and reaction system. So if we look at the whole workflow of the compound synthesis, and of course, we first have to design the route for the chemical synthesis and then execute the chemical reaction. And afterwards, we do the compound testing and analysis on the analytical instruments. Usually, the most commonly used instruments are HPLC, LC-MS, and GC. And if you look at the whole workflow, usually the scientists spend more than 50% of the time on the chemical synthesis. So this is an extremely time-consuming and cumbersome job. And currently, the pain points for the industry, for the pharmaceutical companies, are mainly related with chemical feeding, reaction stage, and also data analysis. So in terms of the chemical feeding, the scientists, the technicians, they have to deal with diversified solid powders, liquids, and various consumables. And this is extremely complicated and cumbersome. And regarding the chemical stage, so the significant challenges include low yields and low producibility. And additionally, many of the reactions are inflammable, explosive, toxic, and hazardous. This may raise the questions for chemical control, for environmental control, and also the safety issues. And for the data analysis, characterizing new compounds, new moleculars requires techniques such as NMR, chromatography, and also sometimes X-ray. So furthermore, the impurity can complicate both structural confirmation and subsequent biological testing. So there are also some workflow challenges. For example, the technicians, they have to execute huge amount of reactions, and usually they only can operate the chemical reaction in a limited space. However, the biggest challenge is that a lot of highly educated doctors and scientists are required to conduct this kind of low profile and tedious job. And unfortunately, this is extremely wasting talents. To solve the industry pain points and challenges, we specifically designed a high throughput, intelligent chemical feeding and reaction system. So our system can conduct microgram grade reaction, and this platform is suitable for common pharmaceutical reactions. And on this slide, I just want to show you the 3D visualization picture, and so that you know the general the structure of this platform. So on the left side, we have the feeding chamber, and on the right side, we have the reaction chamber, and everything is connected through the robotic arm. And so this platform is equipped with 9 independent shakers, and so we can control the temperature from 5 to 150 degrees. And this platform is also compatible with diversified consumables and reaction bottles. And additionally, we can also provide gas protection so that we can generate oxygen-free environments. And lastly, we can also provide replaceable feeding heads. Yeah. And there are also two different models for the chemical synthesis platform. So one model can deal with 8 milliliter system, and in one day we can complete 1,536 reactions. And the second model is able to deal with 40 milliliter system, and in one day we can complete 1,224 reactions. And here I just want to quickly summarize the key advantages. So, for example, the pipetting accuracy of our platform is for 1 microliter, the variation rate is less than 4%. For 10 microliter, the variation rate is less than 2.5%. And the weighting precision is plus minus 0.5 microgram. And additionally, this is also a high throughput platform. So this platform is equipped with 9 independent temperature control shakers, and simultaneously we can conduct 108 reactions. And in addition, so this platform is also equipped with a barcode scanner and imaging modeler so that we can realize sample tracking and whole process monitoring. And lastly, so we can also provide gas protected system so that we can fulfill the oxygen-free feeding and reaction environment, and also diversified consumables could be uh, adopted on this platform. Here, I just want to show you a video so that you will have some further impression about the high throughput platform that we can provide for chemical synthesis and chemical feeding. So besides the hardware support, we can also provide the softwares. So By using our software, we can receive the orders from the LIMS system, and we can also record the data from the whole experiment, and also afterwards upload the data into the LIMS or the ERN system. And additionally, we can also provide information to the scientists for optimizing the experiment design and also select the compounds for the chemical reaction. And here I just want to give you a quick example so that you will know the benefits of our AI agents. So first, we can use the AI agents to identify the potential active compounds which have the high affinity with the potential drug targets. And then we can use the AI agent to design the pathway for the chemical synthesis and also predict the yield of the chemical reaction. And afterwards, we execute the chemical reaction on the fully automated platform, and then we feed the data to the AI agents, and then the AI program can inform the scientists how to optimize the experiment design, how to optimize the route for the chemical synthesis according to the downstream data. So by using our high throughput chemical synthesis platform, we can execute 200 reactions per day, and we can conduct 75 rounds condition screening, and we can increase the working efficiency for more than 40 times. In the second part of my presentation, I want to specifically introduce the organoid automated solutions. And as we know, the organoid 3D disease model have very diversified application scenario. We can use the 3D model for precision medicine, developmental biology, drug discovery, screening, and also regeneration medicine. And in comparison with the conventional animal model and also PDX model, so the patient-derived organoid is very flexible and also cost-effective. However, the current pinpoints for the organoid construction is related with no standardization, low consistency, and also low producibility. And to solve the problem of the organoid construction, we specifically designed several different level of organoid automation. So, for example, we can provide standardized instruments like organoid workstation, high content imaging, and also cell culture incubator. And additionally, we can also build up one-step automation, for example, for the high content imaging and also the drug treatments. And additionally, we can also provide the integrated platform to realize the whole workflow automation. And here I just want to dive into the automations and integrated platforms that we can provide specifically for the organoid application. So this is the first instrument that I want to introduce today is the organoid workstation. And so this platform is equipped with cutting-edge technology. For example, we have 8 independent pipetting channel, and we can provide air pressure sensor, bubble sensor, and also clot sensor, so that we can identify the level of the liquids. And additionally, we also have in-house developed software. This is a easy to use software. It has very intuitive interface, and you don't need a dedicated bioinformatician to use the software. For example, I only have the molecular biology background, and I'm not a software engineer. I don't have the bioinformatic background. I only need 2 or 3 days training to be able to use the software. And additionally, this is also an open platform, so diversified reagents and consumables can be adapted on this platform. And lastly, so we can also integrate many different functional modelers. For example, the cooling modeler, heating and shaking, and tilting modelers for the medium change, and also the centrifuge and positive pressure HEPA system. And with all these different functional modulars, we can cover most of the steps for 2D and 3D cell culture. For example, by using our organoid workstation, we can do matrigel mixing, seeding the organoids, replacing the cell culture medium, passage, and preserve the organoids. Here, I just want to show you a quick video so that you have some impression about our organoid culture workstation. So besides the organoid culture station, we can also provide cell view high content imaging. This is specifically designed for the high throughput drug screening by using the 3D disease model. So this is an all-in-one instrument. So this we can use it as a fully automated microscope, which integrates with the lab cell culture chamber and also the twin focal lens, so that we can generate high resolution 3D picture. And in terms of the light source, we have the colored bright field, and we also have five fluorescence channels. So PE, DAPI, fits APC, and plus infrared laser channel. And additionally, we can also do the Z-stack imaging so that we can scan the samples layer by layer, and afterwards put all the pictures together, generate 3D picture. And one of our advantage is the large field of view. So for example, as you can see on this slide, and the picture in the right frame, this is the picture size from our competitor. And in comparison with our competitor, the FOV from our cell view imaging instrument is 20% bigger. And as I mentioned before, this instrument is also equipped with infrared laser channel, so that besides the imaging-based software focus, we can also do the laser-based hardware focus, so that we can improve the imaging speed and also prevent the phototoxicity from happening. And additionally, we also have in-house developed data analysis software. So by using our software, we can do the multi-field image stitching, cell segmentation, biomarker analysis, and also 3D reconstruction. And most importantly, we can integrate the Optimoid workstation and also high-content imaging, as well as our Optimoid incubator into an integrated platform so that we can realize the whole workflow automation. And the core function of this platform is optimized culture and packaging, and we can also use it to collect the imaging data to conduct the drug sensitivity testing. The throughput of this platform is 4 tissue samples per batch, and in one day we can process 12 tissue samples. And this is the workflow that I want to show you, and we can use it as a demo case. So we first collected a patient sample from the hospital, and we first do the tissue dissociation and then load the sample on the fully automated platform. So we can do the optimize packaging, the preservation, and afterwards we can also use the high content imaging to acquire the imaging data. So by using our software, we can do the octoids counting and also calculate the area of the octoids, and we can also do the IC50 drug sensitivity testing. And in the next part of my presentation, I want to specifically introduce the pharmaceutical quality control solutions. And as we know, the pharmaceutical companies' quality control process is extremely complex. So there are so many departments are involved into this process. For example, the warehouse, QC lab, QA lab, and we do many different chemical testing, biological testing, and dissolution analysis. And of course, the digitalization software is very important to improve the working efficiency and deal with a huge amount of data. And the automations also play crucial role to increase the working efficiency, reduce the loss. And on the top of this, the artificial intelligence program is very important. So in any case, if something happens within this complicated workflow, and the AI program can immediately identify the problem and inform the scientists how to solve the issue. And the most commonly used analytical instrument in the QC inspection process is the HPLC, GC, and LC mass. All these are chromatography analysis instruments. And when you look at the workflow for the chromatography analysis, so we first have to prepare the samples, and then we prepare the solutions, for example, the mobile phase solution, blank solution, and also sensitivity solution. And afterwards, we load the sample on the analytical instruments, and then we load the results into the ERN and IMS system. And if you look at the time that we spent on all these different steps, we spent more than 65% of the time for the sample pretreatment, for example, the mobile phase preparation. And unfortunately, this is extremely cumbersome and low profile job. However, we use a lot of highly educated technicians for this tedious task. And to improve the working efficiency and reduce the cost for the mobile phase preparation, we specifically designed a fully automated platform for it. And here, this is the 3D picture for this platform. So on the left side, we have the solid powder storage, reagent storage room. And here we have a pH meter, we have the filtration modular, ultrasonic degassing modular, and also the powder dispensing modular. And everything is connected through this 6-axis robotic arm. This is the general workflow for the mobile phase preparation platform. So we first manually load the reagent and powders, and then the platform will weight the solid powder, add the liquid into the bottle, and dissolve the solid powder by stirring. And then we can also measure the pH value, do the reagent dilution, vacuum filtration, and ultrasonic degassing. And afterwards, we can label the bottles and also sub-packaging the reagent. Here, I just want to show you a video so that you have impression for our mobile phase preparation platform. And in the next slides, I just want to summarize the advantages of our mobile phase preparation platform. So first of all, this platform is equipped with nitrogen chamber that protects the solid powder dosing system from impact of water and oxygen. And secondly, we have very user-friendly designing. For example, the left side is the storage room for the reagent and liquid and solid powder. So we can separate the cart from the platform so that the technicians can load the solid powder and reagent manually and then put the cart back into the platform. And Additionally, we can also offer the daily inspection process that facilitates audit tracking, including balance and pitch daily inspection processes. And furthermore, we can also provide preventive maintenance reminders, including consumable lifetime, uh, life alarms, and also reagents shelf life alarms. And because some of the regions has very high acidity and basicity level, so we also verify the key raw materials so that we can prevent extraction from happening. And this is a side-by-side comparison between our mobile-based platform and our competitors. So on our platform, we can prepare maximum 40-liter reagents, and additionally, we can put 9 different solid powders and 12 different liquids on this platform. And moreover, this platform is also equipped with 18 channels pipetting system and filtration modular and also ultrasonic uh, uh, degassing modular. And we can also fulfill the regulatory compliance for computer system validation, CSV. And within our company, we also have diversified checkpoint controls. So, for example, before, within, and after the instrument building, we have different checkpoints, SOPs to guarantee the performance and quality of this platform. And lastly, we also have regulatory compliance from NMPA, FDA, EMA, and also many professional organizations. In the last part of my presentation, I want to introduce the intelligent chromatography testing system. And first of all, I want to introduce the AGV robot. So this robot is equipped with 6-axis robotic arm. And on this robotic arm, we also have the door opener, the gripper, and also visualization system. And this platform also can be used for diversified consumables. And by using the AGV robot, we can literally connect different functional units and even different functional labs. For example, the bell bank, sample bank, the sample preparation, mobile phase preparation platforms, as well as the analytical instruments. And here, I just want to show you a quick video so that you have impression about the um, intelligent chromatography testing lab that we established together with our strategic partner, Agilent. Yeah, here I just want to remind you we have experience for dealing many different chemicals. For example, the high viscosity, high corrosive and hazardous chemicals. And additionally, we also have experience for connecting with analytical instruments from many different suppliers. For example, Metro, Thermo Fisher, Agilent, PerkinElmer. And that brings me to the end of my part of representation. So within my part, I specifically introduce the automations, the automated solutions from our company. However, if you want to provide a fully automated platform, integrated platform, the software is also important. So in the next part of this webinar, I will transfer the gear to my colleague, Dr. Xu, and so that she will specifically introduce the software solutions that we can provide. Thank you very much for your attention.
Good morning, everyone. Thank you for attending the seminar, and I really appreciate your time. It's my honor to introduce our digital platform, Revolution, to all of you. My name is Yuyan, and I'm an application scientist at Galatech. My job is to bridge the gap between biopharmaceutical research and the industry application. So today, first of all, I will do a brief introduction about why we want to develop the Billion digital platform and give you an overall picture about the background of the Billion. As you may know that a lot of scientists today, especially in academia, they may still keep a traditional way of doing research that rely a lot on the paper notebooks. In addition, pipetting, logging data by hand, it's tedious, error-prone, and frankly, I really think it's a waste of smart people's time. In addition, our data is in silos, is trapped in silos. Honestly, it's really time-consuming to find one critical experiment from a couple months ago, even written by yourself, and all this make research slow and inefficient. In a harsh reality of R&D in pharmaceutical companies today is that we may spend over 10 years and $2.5 billion in developing one new drug, but many of them may still fail in the clinical trials. This is very disappointing because over 70% of the experiments cannot be repeated, and one of the main reasons is that our scientists have to spend over 40% of their time on manual data management and administrative tasks. Therefore, this is an urgent issue and we need to fix it. And I believe we all know the solution to use AI and automation. And AI is now reality. It's really shaking things up. To fully release AI's potential, we cannot just speed up one piece of the process. We need to connect the entire research workflow into one seamless system. And what we want to achieve in Revolution is to use AI to drive every key component of research. So once we have some great idea to test, we can use AI to predict the experimental designs, and we can choose one of the best to test. And to test this idea, we can input natural language to AI, and it can help us to generate machinery languages which can be operated by laboratory instruments. Once the experiment is done, all the data generated by instruments can be collected and analyzed as the way you want. Last but not least, AI can generate an intelligent report which can allow us to have a real-time feedback and can drive new insights to test. This is Revolution, an AI-driven intelligent software. It contains 8 main modules, from registry module, which contains comprehensive map of lab data to allow track samples full life cycle information, to smart automation scheduling and workflow management. We also have data center and analytics to generate experimental reports and data analysis. So this is Revolution, which is automated, integrated, and intelligent. It connects wet lab work and dry lab analysis to a unified workflow. And here, we are establishing an AI-driven end-to-end research system. This innovative digital platform will change the future of research with extremely high efficiency. So next part, I will introduce a few important modules in Labillen. So the first module is automation. This is a system that involves both hardware, such as automated liquid handlers, and software, such as AI algorithms, to minimize human intervention and improve work efficiency. So the automation is easy to integrate with LIMS or other systems and for workflow coordination. In addition, our automation system uses Silo 2 standard for discovering and controlling automation instruments and supports data reporting for structured and unstructured instrument data. And so far, we are able to connect with more than 200 third-party devices, including the most commonly used devices like microplate reader, flow cytometry, qPCR machine, and a mass spec instrument. And we are very flexible to connect different instruments according to our customers' needs. Our Labillen automation is easy to design workflow. The user just needs to drag and drop features for process design and set up loops or conditional logical functions. And for each feature here, you just need to double-click and set method as you need. Our system could run the workflow automatically and identify logical errors simultaneously. And this Labillen automation allows multiple users to log in at the same time and support multiple devices, data querying and monitoring, as well as collaborative process design among multiple users. You could just log in anytime, anywhere using Windows, Mac OS, or Linux system. Besides the basic feature of workflow design, automation has built-in intelligent capabilities such as the work-cell lab wheel visualization. You are able to visualize all lab wheels and its status. Besides, we have video monitoring so that users could monitor all the workstations remotely. And here we have 4 workstations, and you are able to visualize all the status of these workstations. Lastly, we have integrated AI assistant. Users could chat with our AI assistant, ask questions for help. For example, in this demo, we can type QPEX how to calibrate the video camera, and our AI assistant could provide detailed methods for camera calibration. And our AI assisted search could retrieve all the results that containing the keywords you entered and help you find some specific information you need. And let's see this demo. In this demo here, we could type some keywords to do some search. Here we just type, help me find 48 deep well plates that are not expired. That's what we type in, and we just click search. And then it takes some time to do the search, and then it will return the results that fulfill the keywords that we entered. And here we find 2 plates and 248 deep well plates that are not expired. And that's all about our automation. And the next part, I will introduce how our automation module meets the FDA compliance. So I will go through all the details one by one. First, our automation, it provides customized user roles. And here we can define our users as student, technician, scientist, engineer, administrator, and other users. And for each role here, we are able to give them different permissions to different modules. Here, in the automation, we also have built-in process and method release workflow. We are able to customize different approval workflow with a number of approval conditions, and you can assign reviewers, and we can also have electronic signature for approval. We also support historical version tracking for process and methods. All released files cannot be modified directly. It has to be reverted to draft before editing, and all the modified files can be saved as historical versions. So for full historical versions, they are all viewable but cannot be modified, and all the version records cannot be deleted directly. So we also support audit log query. So we will record all the changes, including user permission, system configuration, device settings, labware attributes, and process and methods. Lastly, we support historical version tracking for process and methods. Here, we record every run, and we will fully display the user interface at the end of each run. We will also display runtime controller, ground chart, operation history, warnings and errors, as well as all the variables. In next part, I will introduce a few other modules in the Labillion. So first, it's the registry model. So for registry, it can provide a traceable process with standardized data processing and structured storage. It also provides knowledge graph-based sample mapping. So for registry, we are able to generate model configuration using schema. So what is schema? It's a standard way that we can organize and store our data in a structured manner. And in our registry, we provide all different types of schema like samples, reagents, consumables, equipment, project, electronic notebook, and other customized materials. Because we provide different types of schema, we are able to avoid data confusion and conflicts. Because all the data are saved in a structured manner, we are able to retrieve our data easily. So to create schema, we just need to click this blue button, and then we can import our schema name, key value, and set some properties of the schema. For example, if we just created a schema for sample named protein, we are able to generate it and then click registry. We just need to click the schema we created for protein and then import the protein name, which project is affiliated, and the properties for this protein. In that way, we are able to register this protein sample. And another feature in our registry is it provides knowledge graph-based sample mapping. So this is very helpful. For example, if there are several team members work together to produce some protein and do some modification, team member A may just create a plasmid, and team member B can use this plasmid to express in the cells and to produce some protein. And team member C, he can use this protein to do some modification and purification. And if you want to trace where does this which plasmid we use to generate protein and which kind of purification we use, if we want to trace all the samples, we are able to generate schema and link them to each other. What we need to do is when we create the schema, when we create the samples, we just need to link this protein to a schema called plasmid. In that way, we can generate a knowledge graph-based sample, and we are able to view where does this sample protein come from and what is this sample protein used for. So once we have registered all our materials or our samples, reagents, we are able to do inventory management. Here, for our inventory module, it's easy for operation, and we provide end-to-end tracking with smart alerts for low stock and for materials that are about to expire. We also provide real-time visualization to view all the equipments that registered in our labs. So in our inventory management, we have company level and laboratory levels. So for company level, it's a larger scale for all samples, reagents, consumables that are stored in the company. We're able to do management and view the details. And for laboratory level, it's in a smaller scale. For one company, it may contain several separate and independent labs. We are able to register these labs and equipments, materials in that labs, and view all the details for those inventory. So for company level, we are able to do inbound and outbound. Suppose you just order some reagents and some consumables from a supplier and we just received these materials. We are able to do inbound and store all the information for our materials. We can record batch number, storage location, quantity, project affiliation, and expiration date, and so on. But if you want to transfer your material to some other labs, we are able to record the information. We can just click outbound and we are able to record the quantity and which recipient will receive this material. Besides, we also provide inventory details. You are able to view the material. You are able to view the operator, barcode, quantity, storage location for each material that you registered. And if you want to invalidate your materials because of it's about to expire or it's no longer exists, you are able to do an operation called invalidate. We also provide another operation called aliquot if you want to put your samples into smaller portions and use them for different experiments. For our inventory system, we also provide a few different management. For example, we have low stock management. If you want to receive alerts when certain materials are lower than a threshold you set, you can just import a material threshold and you will receive a notification in your message center like this. We also provide expiration management, so you can set how long before the expiration date you want to receive an alert like this. And we also have records for relocation. We will write down the material and we will write down the location before and after shift, time and operator. We also have invalidation records. We can save time, reason, and operator for invalidation. And for laboratory level inventory management, it's almost the same as a company level, but in addition, we also provide laboratory registration and machine registration. In that way, we are able to register all the labs and all the machines in that labs so that we can visualize all the laboratory and equipment information. For example, if you have a lab and a workstation like this, we are able to register all the freezers and equipments, PCR machines, all different machines in this workstation. And here we have a -40 freezer. If you want to view all the materials stored in this freezer, you just need to click this freezer and we are able to see a window like this. Here we have blue and white space. So blue means it's filled with samples, and blue and white means it's not filled. So if you want to see all the material information in the freezer, we just need to click the space, and here we just click the third shelf, and we are able to view all the plates, boxes that are stored in this freezer. And for the first plate here, we are able to see a 96 well plate, and we are able to click each well of the plate to view the material details, including the name, barcode, batch number, type of the material, quantity, expiration date, and the status of this material. In this way, we are able to have a real-time visualization for all the materials and all equipments that registered under certain laboratory. So that's all about our registry and inventory, and we also have electronic notebook in our Levelland system. So for our notebook, we provide customized notebook template, and you can use this template to create any notebook you want. So for our notebook, we provide a lot of functions, including editing tags. You can add whatever tags you want, and we can insert dates, tables, plates, text box. We are able to upload attachments, and you can preview those attachments. We can print the notebook or save them as a PDF file. we can check the version history and reset the notebook to an older version if you want to restore some older data or notebooks. And we can view the timestamp to see what time and what modification you made for those notebook. And we can also write this notebook with a few different team members, and you can add your team member to remind them or to assign them with different experiments through message notification. We can also submit the notebook when we finish it, and we can assign reviewers to do approval. We can export the audit log to view all the changes we made in the notebook. Here I just list some of the functions in our notebook, but we do have more. I might be able to introduce all of them, but next I will introduce some of the advanced components in our notebook. We have a lookup table. Here we are able to view all the sample information and track them in our lookup table. Here we have some 96 wheel plates, and if you click any of the wheel of this plate, we are able to view the information stored in this wheel, and we can see where does this sample come from, and what is this sample used for, and where is it stored. We also have a smart chart. For example, if we have some gene expression for control samples and treatment samples, and if we want to make a heat map to show the gene expression for different samples, we can click this smart chart button, and we can just use our natural language to the integrated AI. We can type, please use name as columns and expression level as number, and help me make a heat map that is clustered by gene. And our integrated AI can generate a heat map to show the gene expression in different samples like this. Lastly, we have AI table reading. So for all manually recorded tables, we can just take a picture and upload this picture in our notebook. We are able to recognize this table as an online digital table. In that way, it can save time for us. We don't have to manually record all these numbers and all this data. So that's all for our Labellian notebook. Thank you for your attention, and please let me know if you have any questions.
Thank you both for your informative presentations. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just type your question into the Q&A box and click submit. We'll answer as many of your questions as we have time for. Let's go ahead and get started. Our first question is for Dr. Xu. This question asks, what is the core competitiveness of Labellion digital platform?
Yeah. Hi, can you hear me?
Yes.
Can you hear me, Casey?
Yep.
Yes, great. So, thank you for your question. So I think the core of our competitive advantages is that we have moved beyond simple digitalization to create a truly intelligent laboratory ecosystem. So I think while many of the platforms may offer digital tools or automation, we do have integrated AI at the very heart of our automation or digital tools. So I think first, our AI doesn't just execute some predefined workflows. It can also help us to analyze historical and real-time data, helps us custom query and data searching, and also provide software support. And secondly, I think our AI capacities can extend beyond preset process. So this automation system we have that features AI API-driven connectivity to instrument outputs. It can enable automatic data capture and ingestion for subsequent processing. And lastly, I think our platform meets with the FDA compliance and provides the necessary features such as approval workflow, historical version tracking, audit log, and so on. So based on that, I think that's all our advantages over our competitors.
Thank you so much. All right. Our next question, and this one's a little bit long, so bear with me. This one is for you, Yicheng. What happens when a mistake occurs during the mobile phase preparation organoid chemical synthesis workstation? For example, clash of the robot with interior. Do these robots have any emergency procedure included to interrupt the workflow? And how easy is restarting the workflow? Or does one have to start the entire workflow from the beginning?
This is a very good question. So first of all, I just want to mention, so our platform can run multiple workflows at the same time. So in any case, if anything happens for one workflow, the system will reschedule the other experiments workflows so that the error won't affect the results of the other experiments. And if the problem or the error isn't so critical, so we can also solve the problem and restart the workflow. So everything can be solved very easily. Yeah, that's my answer for this question. Thank you.
Wonderful, thank you. Okay, our next question back to you, Dr. Xu. What deployment modes do you provide? And Dr. Xu, I'm so sorry, your microphone is muted.
I'm sorry. So currently we provide two kinds of deployment. So we provide on-premise solution and cloud-based SaaS, which is short for software as a service. So it's totally based on our customers' needs. So if you want to install the software on your own server and within your own organization, we can use on-premise. In that way, you can have higher control over your data, but it might have higher costs. And for the second solution, for the SaaS, I think this one is more common right now. So you can access the platform as a service over the internet, and then we can host and manage everything on our secure. So what you can do is you can, and we can also provide regular maintenance of the software, so it might be easier for the user to use.
Wonderful, thank you.
Yeah, I think that's it. Yeah.
Okay, we're going to go back over to you, Yicheng, for this next question. This one is asking, do all of your showcased devices have an FDA?
Yeah, so our software has the FDA 21 CFR 11 certificate. And besides this, we can also provide the regulatory compliance for the FDA, NMPA, and EMA. So this is the answer for your question. Yeah, thank you.
Wonderful, thank you. And we have time to squeeze in just one more question. This next and last question asks, how does a cell workstation improve workflow efficiency and productivity in a manufacturing lab environment?
Yeah, I think I will take the chance to answer this question. So actually, this is very good question, and of course, a lot of people talking about the automation, the integrated platforms, and they always think about the working efficiency and also the throughput. However, this is not the only advantage when you are using the automations and integrated platforms. Specifically, when talking about the integrated solution, so the biggest bottlenecks or the pinpoints in the industry or in the research field are the low consistency, no standardization, and also low reproducibility. So by using the cell, the optimized workstation, we can solve the problem. So I would say that's the biggest benefit for the cell culture workstation. Yeah, to improve the reproducibility and consistency. Yeah, thank you. That's my answer for this question.
Wonderful. I want to thank you both for your time today and your important research. We would also like to thank LabRoots and our sponsor, Galatech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.